So I am going to now speak on this topic, research projects, how to develop and execute them. I will not certainly bother you with the execution part of it, but we will perhaps share some ideas about how we can develop new research projects. May I know if any of you are married other than the staff people here among the students? ಯಾಕಪ್ಪ ಮದುವೆ ಅಂದ್ರೆ ನಗ್ತಾ ಇದ್ದೀರಲ್ರಿ ನಾಚಿಕೆನಾ? Any of you are married here? No, yeah, she is married. Thank you, madam. Your name? Pardon me? Deepa. You have a lot of problems now. Okay, you all know about the pollinators and flowers, isn't it? Right? Right or no? If you don't respond, I will not go forward. So you all know about the pollinators and the flowers, and what is very simple about them. If there is a flower which is colorful and attractive, bees visit to that, isn't it? And why do bees visit to that? They visit because flowers offer honey and pollen. They are also colorful and they show a lot of designs. And so inevitably, bees have an instinct to now be attracted for this honey, pollen and colorful flowers, isn't it? Right? So, in other words, because it is instinctive for the bees to be attracted by the flowers, whenever there is a flower, bees should now generally go there. Right? Any questions on that? But have you ever questioned why is that bees don't visit Aishwarya Rai when she is wearing a garland like this? Aren't there flowers there? Aren't there flowers in the garland? But why do not the bees visit there? Pardon? The fellow who made garland never removed the pollen grains. Yeah, it's not covered. You see, so many flowers there which are open. Right? That's why I asked you, madam. Did any bee bite you when you were marrying? I'm sure you had a garland on your neck. I'm sure she's also married. Yeah. So you never were afraid about the bees coming, isn't it? If at all bees come to the garland, no bride and bridegroom will wear flowers or garlands. They will throw them up, even if it is a rule, isn't it? In other words, you see that despite the fact that bees have an instinct to visit the flowers, we don't see bees coming to the marriage halls. Am I clear? You all agree with that? Have you ever questioned why? Marriage? That's for us, not for bees. Marriage tension never questioned later, but later you would have seen so many others' marriages where you are not tense, you wear more beautiful saree than this and go there. Yeah, so in other words, even when we are now looking at so many marriages, pardon me? Flowers are plucked. Madam, bees will not know where the flower is, right? Whether you have a sweet served along with the cup or on a saucer, we eat the sweet, isn't it? No, not all flowers are washed. They are simply harvested from the field and they are now simply over. Ashtond time illideyamma, nuraru haragalu beku ಒಂದು ಮದುವೆನಲ್ಲಿ. Any answers? Incidentally, this is precisely what, there is some problem. This is precisely what one scientist asked this question. What is that? Why bees do not visit garlands? And that person is Dr. Y. Rajshekar from Animal Resources Program, ISBD, Imphal, Manipur. Isn't that a question? Have you ever bothered about it? You seem to be very reluctant to say yes or no. Isn't it a question? It is a question, isn't it? In other words, there are so many things that we can question about our surroundings among our living styles, and that is exactly what Rajshekar questioned. What he also did was the following. The moment he asked this question, he had something in his mind, and so he went about imagining that this could be the reason, and so set up an experiment. The experiment is very simple. Look at the two kinds of flowers, one on the left hand side and one on the right hand side. He set it up in the open field and sat in front of them and started looking at whether bees come or not. Surprisingly, he did see that bees come. But where? Not to the left side, but to the right side. Why? I am asking you, you should now tell me why. Do you see any difference? Pardon me? No, he did not spray any chemicals. There's just those kind of garlands on the left and those kind of garlands on the right. He found that they did not come to the right side, left side, no? Left side batch, and they came only to the right side batch. Yeah. Who told that? Give her a big clap. See, that is what is required in science. Keen observation. Rajshekhar did a keen observation. What is that? Bees should come to the flowers, but they do not come to the garlands. And he assumed something, and he tested this assumption, I'll come to, and he found that when he set up this experiment, they did not come here and they came here. The difference is there is a greenness on the left-hand side. Does it mean that they hate green? If they hate green, they can never come to the plants, isn't it? Incidentally, let me go forward. The green thing that you see there is this. What is that? Any idea? You might have all smelled it, is it? Yeah, it is called davana. It's called davana in Hindi and manje patre or urige patre in Kannada, and that is the leaf that is put with all the garlands. We think it is actually for smell, could be, but for putting smell, there are so many different kinds of flowers or different things, but they put only this because this has a function. And Rajshekhar thought that this could perhaps be a repellent of the bees. So when you put this green leaf along with, that is davana, along with the flowers, though it is very much nicely smelling to all of us, bees do not like that smell. In fact, not only that, there are several species of this davana. The genus is Artemisia, Artemisia vulgaris, Artemisia annua. There are about 12 to 13 species that people generally use, and most of them are actually also used as medicinal plants. And what is more, in Karnataka and in South India, Artemisia vulgaris leaves are used in garlands. It acts as a repellent, and in this particular species, Artemisia, for the first time, an alkaloid called artemisinin was extracted. What is more interesting? One among those Artemisia species also gave an anti-malarial drug. Rajshekhar extracted this artemisinin from Artemisia and found that it can even kill cockroaches. It also repels insects. Sometime during 2015, a Chinese lady got Nobel Prize for having worked on this plant, because she now found that in their traditional knowledge, this plant was being used for some medicinal purpose. She chased it and discovered the malarial drug, and she got Nobel Prize for that. In northeastern region, they use this as a sedative for catching fishes. You extract it and put with small worms, and when the fish catches it, they become sedated and they can easily catch them by the hand. Isn't that interesting? You don't want to give a big clap to Rajshekhar? Be little responsive to some of these things. Such a beautiful work, isn't it? Incidentally, the reason why I am talking about this is the following. We are working on how to write projects, how to prepare research projects. Generally, I see that lot of teachers go to the library, look at, nanochemicals or nanoparticles is doing lot of work. They will come back to Surana College, Madam Principal, and tell, Madam, I am going to write a nanoparticle research, Madam. It's not essential that we need to now follow or copy someone else. There is so much that around us. In fact, the most important thing that perhaps we all need to now learn is that observe the world around us, look for surprises that are generally taken as very obvious by others, but actually not obvious. What is obvious in the garland thing? It is obvious that bees don't come there. But the most surprising thing is they should have come there. That is the kind of unobvious things that have become obvious in our mind. We should now perhaps recognize them, and there is no need to borrow ideas from other scientists. There is a syndrome in India that whatever is being done by Thomson or Johnson or Nicholson from USA is perhaps good, let us follow that. No, no need. Rajshekhar did not follow any Westerner. He only saw the Indian culture, and from the Indian culture he raised a question, and from that question he now tried to answer. And he now came out with a fantastic project. DST gave them something like 25 lakhs for the first time. He's still continuing on that work. In other words, having observed the world around us, we need to perhaps now identify a question and think of a meaningful question, just not a question. I would say meaningful question because only when we raise a meaningful question, perhaps we can go ahead. So, prescription number 1, like a doctor, I would like to give you all, if at all you want to now write a research project is, first of all, you have to raise a question. You all agree with me? The first prescription is raise a question or ask a question. And how do you now ask a question? In fact, everything, whether it is arts or science, everything begins with a question. If you don't have a question, there is nothing to now investigate. If you don't have a question, there is no road to go ahead. So keep your eyes open and observe the world around. And number 2, you may get several questions from that. Some of them may be ridiculous, some of them may be very meaningful. So filter out those that are very meaningful. If you don't identify a question, nothing happens without that. And so you have to now put in any amount of effort to now observe these unobvious things in the world surrounding you, raise several questions, among them identify the ones that are very meaningful. In other words, ask a good question of the kind that Rajshekhar has asked. For example, why do plants have laxatives? You know what is a laxative? If you have trouble in the bowel movement, if you can't go easily for this in the morning, they give you this laxative. You take, maybe coming in the form of the paste or seeds or whatever it is, you take that and automatically bowels move and you are relieved of your troubles. You know what I mean? That is laxative. And this laxative, which is actually inducing bowel movement in human beings, is there in the plant. What is it doing there? Why should plants have laxatives? Right? Why should, for that matter, why only laxative? Why should plants have medicinal plants? You know, when some plant extracts are taken, our headache is gone. Plant doesn't have a headache, only we have a headache. Why is that chemical is there in the plant that cures our headache? Have you ever questioned this? So, questions of this perhaps are the ones that we need to now raise. How do ants find sugar in the, how do ants find sugar box in the kitchen? You know, there are hundreds of boxes in the kitchen, isn't it? If your mother or yourself, mother will keep it correctly. If you keep the sugar box with little open, lid, automatically next day morning ants will be there. Out of hundreds of boxes, how do ants find only the sugar box? Have you ever questioned? Madam, have you ever questioned? It's a problem in your house, in your kitchen. Have you? Why? And that could be a question. There was a small, girl, I think of, in fact, none of you are as young as this girl that I'm talking about, who came to me for an academic summer fellowship. She raised this question. I generally promote people to now, I develop their own questions and ask their own questions. She told, sir, 2 days she took to now think about. She came back with a lot of shy and told, I think Sachin knows her, that northern girl. Okay, whatever her name is. She came, sir, I have a question, but I don't know whether it's right or wrong. My dear, tell me what is the question. Sir, I have found that in my kitchen, always ants find a sugar box. How do they find it out, sir? I told, very good. Very good. Because there are so many boxes, how is that ants find only the sugar box? There could be 2 reasons. One is, they will be randomly moving, and if at all they find sugar box, they now attack them. Possible? Other, they can smell the sugar. Can we smell the sugar? We can't. Maybe ants will smell the sugar. So I told, very good, go ahead and do it. She simply did the following kind of experiment. What is that? She kept cotton with the sugar in one place, cotton without sugar in another place, sugar solution, okay? Just the normal water and the sugar. And she went on about 5 meters away from each other. She went on observing, where do the ants come first? If they are randomly moving, they should come equal number of times to the sugar swab, but also equal number of times to the cotton swab. She found that in about 100 trials she did, 80% of the times ants attacked the sugar swab more often and faster than going to the ordinary swab. That means ants are capable of smelling. Isn't that a good question and a good answer? Now we can ask further, how are they smelling? By the antenna or by something else, or do they have something in their leg? Things can continue. So in other words, asking a good question is very important. How does the water lily stay dry? You know water lilies, isn't it? The leaves of water lily never get wet. If at all you put water, it just drops up. A biophysicist in America took this study. He was not a biologist, he is a biophysicist. He took up this study and came out with a fantastic work. We will not perhaps go into the detail. So in other words, asking good question is the beginning of all the research. If you don't have a question, I think you cannot now do research at all. I am stressing this point of if you don't have a question, you cannot do research, because I see a syndrome sometimes. Lot of people come to me with this kind of questions. What is that? Analyzing phytochemistry of medicinal plants of Karnataka. Sir, I want to analyze the phytochemistry of medicinal plants of Karnataka, sir. Which medicinal plants? There are 800 medicinal plants. Which phytochemistry? No, they will not have any direction. I think that to me is not a question. Now, please read the 2 lines before that. What is that? The question you are asking should be simple and meaningful. Only then perhaps you can go ahead. And you should not be broad and open-ended. These are open-ended questions. Analyzing phytochemistry of medicinal plants of Karnataka. That is not going to take you anywhere, because there are so many medicinal plants, so many chemicals. What are you going to achieve with that? Some people come and say, I want to study the endangered plants of Western Ghats. Very good plants. There are some endangered plants. You want to study them. For what purpose? And for why? So only when you have a question of what, why and how, you can perhaps start writing a research project or can start doing, not with these kind of open questions. Some people have another syndrome. The syndrome is, sir, biotechnology is on the lead, sir. I want to work with DNA. What is that you want to work with DNA? Anything on DNA, sir. You might have seen this kind of, curiosity among the kids. That is not going to lead you anywhere. Let me give an example of how, in fact, one event happened with a girl who came to our lab. I don't know if Rajini knows about this girl. This girl called Ganeshaiah came to our lab. My colleague, Dr. Umashankar, who is the major advisor for Rajini sitting here, he was supposed to be the mentor for her, guide for her. She came to him and she told, she's from Coimbatore, very smart lady, very nice lady, and she was very intelligent, but corrupted by this view that anyone who does on DNA is doing a good work. So she came and asked him, sir, I want to work on DNA, sir. Our lab, of course, was working with DNA, but we don't work on DNA because we want to work on DNA. There should be a question for us. So she told, I want to work with DNA. Umashankar asked her, what in DNA? Anything, sir. Literally, she told like this. Umashankar told, no, in our lab, we generally don't work like that. We ask a question. You come out with a question, maybe then we can work on DNA. She had no answer. In our lab, as Sachin and Rajini and others would know, or she also would know, we generally go out once in a month or once in 2 months for a kind of field trip. So incidentally, during that week, our team was going to a field trip. So Umashankar told Ganeshaiah, you join us also. We went. We went to, Kokrebellur. We went to Kokrebellur. We were observing these painted storks there. She also was with us. We spent about 2 to 3 hours and suddenly she found something very exciting. What is that? She found that some of these painted storks were getting twigs in their mouth to their nest. They go out, foray somewhere, get the food, but along with the food, some of them were also getting these twigs. She asked Umashankar, sir, why are they carrying the twigs? No one knew. Why are they carrying the twigs? The question why is perhaps the most obvious thing being seen by millions of people there, that they carry the twig. But this question, why, was not asked so far. And so she asked, Uma Shankar was excited. Ganapriya, you can work on that. Is it related to DNA? But isn't that a question that you would like to answer? So she now took the binocular and went on observing, and she found, why do they get twigs? Generally, when you now start observing them, you see that when the twig is brought, it's broken into pieces and they prepare bed out of this twig. For whom? For the fledglings, the babies that they lay there. So they replace the bed almost every day. They throw out the old sticks and they now break this new one, not almost, but let us say very regularly. So they replace this almost every day and they use these plant twigs, fresh plant twigs. But the question is, which plant twigs they get? Number one, why? Number two, which one? Of course, you can simply see through binocular that they are replacing the bed, that is the old prepared bed, and throw them and prepare a new one. You can say, yeah, they use the plant twigs to prepare the bed. But why throw every day? Why can't you continue to have them? Number one. And number two, which plant are they using for preparing the bed? So these are the questions she pursued. In other words, you see that when you ask a question, a door is open for several things. She simply saw the twigs being carried and asked the question why, and that led to further questions. In fact, I would also say that if you have identified a good question, half of the job is done in our research. I will tell you what to be done for the other half little later. So what she did was, she now developed an hypothesis. What is the hypothesis? They bring them for preparing the bed. We don't know why they are throwing out. We will come to that little later. But let us ask the question of what kind of twig they get. One hypothesis is that they don't bother what twig. Wherever they go, they break a twig and come back and use it. One possibility. That is what you call null hypothesis. Or, no, they are using twigs from a specific tree or a specific plant. That is the other alternative. And then, if yes, what is yes? If at all they are using of a particular plant, why are they using only that plant? Why not other plants? Are you all with me? Okay. Perhaps to maintain the hygiene. So we have to answer these questions. What she did was the following. They bring the twigs, they throw out the old ones, collect them and identify what are the twigs they are using. We can't catch hold of from their mouth, from their, when they're their plate. But anyway, they are going to now throw them out, the old ones, collect all of them and identify. It turned out that out of 128 such sample set that she got, 65 belong to the plant Thespecia papulnia. And about, see the last column, 25 belong to Delonex elata. And about 7 belong to Ficus bengalensis, 12 belong to Tamarindus indicus. So she came running to Uma Shankar and told, sir, they have a specific plant to be used, isn't it? Would you all agree with that? They have a specific plant. What is that? Thespecia papulnia. But Uma Shankar is a dirty man. And I'm sure Rajini knows it very well because he was our guide. He wouldn't let anything simply accepted. He told, yes, of course, there are 65 twigs of Thespecia papulnia you have got from the thrown out, nests. But what if Thespecia papulnia is abundantly available in the area and so they are getting Thespecia papulnia randomly, isn't it? In other words, wherever they go, they get a twig. It may so happen that Thespecia papulnia is abundant in the area and so it is abundant in the nest, not that they have a specific, isn't it? So she was asked again. She went back, located the trees all around in 1 kilometer or 2 kilometers surrounding that, and she got the data for that. And here is the data. In the data also, it turned out that Thespecia papulnia is in fact most abundant in that area. But this again won't solve the problem. You have to calculate, if that is the proportion, how many twigs should belong to Thespecia papulnia? Are you all with me? So don't bother about the calculation, that you can learn later what they use chi-square test. And accordingly, when they used chi-square test, she found that Thespecia papulnia is being brought by the birds much more than its availability in the nature. More frequently, they are now preferring to get the species papulinea. There are 97 out of whatever the total number, but if you calculate and do that statistical analysis, it is not that they are randomly getting it, they are particularly, specifically getting twigs of the species papulinea. Are you all with me? Now, one question is answered, that birds have a preference to get only Thespecia papulinea. What is the next question? Why? Why are they getting Thespecia? Does Thespecia papulinea has a smell that these birds like, or is there something else? So, it is a second question that she pursued, that is Thespecia papulinea. Incidentally, why do we now prepare a fresh bed every other day? Why does your mother removes your bed sheet and puts a new bed sheet? Yeah? Neat. What do we mean by neat? Pardon? Hygiene. What kind of hygiene? Very good. Hygiene. We are moving one step. Neat to hygiene. When you say hygiene, if you don't do it, what will be the problem? Yeah? Insects. Who told that? Who told insects? Give her a big clap. In fact, one of the most abundant thing in our bed sheets is what are called mites. You don't realize that. If you don't replace your bed, I mean, not you, if your mother doesn't replace your bed sheet every now and then, third day you will be scratching your hand because of mites. Gajji anta kelidira bandbudutte maigella. And that is nothing but that is nothing but the mites getting into your skin and actually biting it. So, mites are the most frequent visitors to your bed, and these mites will be there not only in your bed sheet, even on the bed sheet prepared by these painted storks. So, she now proposed one hypothesis. If mites are there and if the birds are getting Thespecia papulinea, perhaps Thespecia papulinea might be killing the mites. Is it true? So, she went about and investigated. Don't bother about the details of this experiment, only concentrate on this. Now, this is no Thespecia papulinea, just water extract on mites. This is 10% of Thespecia papulinea. That is, she now takes 10 grams of Thespecia, puts it to 100 ml and crushes it. And this is 20%. And you don't bother about the right side. As you can see, what is there on the Y? Mortality. Mortality is very, very little, less than about 10 or 20% in water, whereas in 10% Thespecia papulinea, 100% mortality in 72 hours. That means Thespecia papulinea stem and twigs and leaves have some chemical that kills what? Mites. Thus, she proved that perhaps these birds are preferring Thespecia papulinea because of their medicinal property. What is more surprising is, when you now start doing these experiments, you also start collecting literature. When she was collecting literature, she found that this is already known to human beings. In fact, in this Siddham website, this Thespecia papulinea is already recommended to be used for what I told Gajji, or for a skin treatment. Put all of them put together, you have proven a new element of science, that birds also know medicinal value of the plants. Isn't that very exciting? Birds also know. See, we did not know. They have somehow found out that Thespecia papulinea kills the mites. That means birds are also capable of doing science as good as you and me. Maybe we are copying from them. Isn't it? In other words, if birds, painted stars could do science, why not you and me? The only thing required is to ask questions when we now look at them. So, I also like to now point out 3 important things from this Ganepriya's experience, that lady who did this work. Number 1, what she did was, not only she asked the question, she developed an argument that this could be the reason why they are now getting Thespecia papulinea. She explored the alternative arguments and hypotheses. And what is for instance, in one among the hypotheses, what she told, birds are simply getting randomly any plant. What is the alternative? No, they are getting only a particular. So, these are alternate arguments, and she tested those arguments. Number 1, raise the question. Number 2, make some arguments. And number 3, test them. Any questions on this so far? Sachin has told me that I should now be talking to you till 11:30. So we have a lot of time. It can be very interactive. If you have any questions, please don't hesitate.
When she found out that it is already available in the literature, was she demotivated?
Actually, fortunately, one should, okay, very good question. Here is a situation where we are all falling into the trap. Let us say for a moment that the moment she raised this question, she went and did the literature and she found that Tespecia populina is already being used for skin treatment. That does not still prove what the bird is doing, right? So there is no need to be demotivated, number 1. Number 2, even if she has now found and has discovered that all that she did with lot of excitement has already been known, never ever be disappointed. You should now get inspiration. What is inspiration? Without that knowledge, you have now discovered something. That means you are capable of discovering several other things also. That's the way you should know. In fact, our question is very exciting and I was very particularly interested on that question because it has happened in our own life several times. We do something exciting. After about 1 year when we are typing or writing the paper, now we see that about 10 years back someone has already done that. It should not disappoint you. You should know promote you. That means independently you have done a work which has been published. That means your work is also worth publishing. That means you are capable of thinking as good as any other scientist, isn't it? And so that is the way. Look at the positive side of any of those. Are you all with me? So in other words, yeah, any other such questions? Boom. If I start continuing that, madam, I will not be able to digest this whole talk. Well, she wrote a paper, number 1. And then, this is only 3 months work. She came here as an intern of the academy, INSA Academy. And one can now continue to do this. I'm very happy she asked this. That means she is now excited about this work. Aren't you all? Yeah. Anyway, look at that. The point here is she came to do work on what? DNA, but she can still do work on DNA. How? Maybe she has to now go back and look at the chemicals available in this Tespecia. What is it doing to mates? How are they killing? So the whole area opened up. Let me also tell you something. This is exactly parallel to another story that happened with a lady called Jane Goodall. Jane Goodall is a lady who was working with the chimpanzees. Some girl went to do PhD with her and this girl similarly observed that chimpanzees were now behaving very oddly on a particular night. Morning 8 o'clock, 3 o'clock, one chimpanzee got up and went out of the nest and went to a particular plant and used the plant, swallowed it and came back and slept. And this girl pursued that and found that that particular leaf of the plant has broad range antibiotics that are killing the worms in the stomach. Now 3 o'clock early in the morning, how can you expect a chimpanzee to get out of its nest and go? Because she observed, she chased that and she found that it is using a medicinal plant. So it's not only birds, chimpanzees also can use medicinal plant. Now what is interesting out of both this chimpanzee story and the bird story is that animals are also capable of discovering medicinal plants. Much more than that. By observing animals, you can discover medicinal plants. That lady discovered medicinal plant by observing chimpanzee. Ganeshaiah discovered medicinal plant by observing what? Painted stalks. So in other words, there is a new field now that is emerging. It's called zoopharmacognosy. Zoo means animals, pharma means medicine, cognosy, discovering medicine through animals. So it's a new field. Billions of dollars are being spent by the pharma companies on students like you. You go and observe monkeys and see what is the special that they are eating other than their food. If they are eating something special, find out why that animal is eating that. Maybe a new medicinal plant will come. So there are these new developments. Though discovery of Ganeshaiah is looks very simple, look at the implications of that, right? And all this started with only one thing, that she started observing that painted stars are getting a twig in her mouth. So like that, Observe the nature surrounding us, raise interesting questions, try to answer them. Fantastic work can be done. So prescription number 2, what was prescription number 1? Raise an interesting question. Prescription number 2 is answer the question. To answer the question, first of all, set up an argument as I told, some hypothesis or some argument, and put them to test. When I say put them to test, there are several ways you can do it. You now logically think whether what you're thinking is correct or not. And number 2, go and look for the literature. If someone has already done it, or if there are indications that this perhaps could be the reason or this argument could be correct. And discuss with your colleagues, because several minds put together on a question is much more valuable than you alone, putting the question to yourself. And accordingly refine the argument and arrive at a testable argument. Once you arrive at a testable argument, you have actually half done the job. I told finding a question is half of the job done in research project. Developing an argument and coming to the level of working is almost half of the other half to be done. If you have come to that stage, you have almost 75% of the work is done. In other words, a good question is half the job done, and if you have a testable theory, half of the other half is achieved. The rest is only putting together some material and then questioning. Any doubts on this? Any queries? Any questions? Yes. Why did you see? Gaps in the research. Isn't that interesting? Why did they bring other twigs also? Incidentally, she found that there are some 3 or 4 species that they preferentially get it, and sometimes, those species can be now tested as whether they also have medicinal plants. Now, why are they getting others also very randomly, very rarely? It might so happen, my hypothesis, I might be wrong. It may so happen that it is now foraging from the morning to evening, and then while flying back, it reminds that, oh, today I have to clean the nest. This species is not available. Some fresh twig has to be brought. Could be. Like now and then, we now use some bed sheets which are not so beautiful, but also, for the sake of keeping the nest fresh and clean, they might be getting, when they don't get Thespecia, on their flight back or so. It could be, I'm not sure. But the fact remains that there are some other tree species also, other than Thespecia, which are also equally preferred after Thespecia. Any other questions? You see how questions start coming out? Yeah. 1 minute, madam. Yeah. Yes, they could have used it. Very interesting. If anything, remember, if they start removing the same tree, the shade on the nest will go off. So they don't want to damage their own house. They want to get the twigs from outside. Yes, madam. Possible. See, we are now telling Thespecia kills mites, but there are ticks also on that. Maybe ticks are killed by some other plant. We don't know, right? So, you see, one small observation leads to almost 2 to 3 PhDs, 2 to 3 projects. Each of them worth 20 lakhs, 30 lakhs to be returned to DBT and DST. Any other questions? Yes. I'll come to. She's still now on a question that I raised about 10 minutes back. Very interesting, madam. I'm happy you have not left that. I will come back, I will answer you. I will tell you why you are taking laxatives. Shall I go ahead? Yeah. Yes, possible, but we are now looking at this Cochrae builder, where these painted stars are there, where they are using Thespecia. Yeah. Correct. Now there is a third PhD coming now. If the painted star is in some other place and Thespecia is not there, what plant they use? They must be discovering some other plant, isn't it? The third PhD came about. You see, in the last 10 minutes, you have already thrown out 4 projects or 3 projects at least. So each of them can be a project, isn't it? Good question, sir. Very good question. Any other things? Shall I go ahead? Okay. Yes or no? Madam, they are all from your college? I don't think your Sachin or anyone has encouraged them to ask questions at all. Said, listen to what I am telling and go back. Huh?
Sachin should stop asking questions so that my kids can ask questions. So now I know from where he has got this, what do you call,
Okay.
asking questions.
Okay. So please respond. If you don't respond, I will think you are sleeping. No? Okay. Shall I go ahead? Yes. Yeah. Yes. Yes. And now, prescription number 3. You have to now do the experiment. Number 1, question. Number 2, develop the argument. Number 3, experiment. Now, in the experiment, test the argument, but while testing the argument, you have to work out the methodology neatly. And it's here that you need some science training. When I say science training, you need to now get into reading papers, understanding how science is done, how experiments are set up. So, you also now work on the methodology and feasibility. You can't simply say, I want to work on this and go and ask principal, madam, give me 10 lakh rupees, I want to work on this. You have to work out the details. Okay? And subject material needed. What is the material that I am going to work on? Is the subject material available or not? Experimental protocols to be adapted. Assess requirements, what material, what chemicals I need, and can I do this experiment? Sometimes some chemicals may be very costly, and the question you're asking may not be worth putting all the money into getting that material. So, infrastructure needed, assess the gaps in methodology and requirements. These all will come automatically when you start doing the experiment. But more importantly, at this point, when you're about to do the experiment, you have to evaluate. Can I write a proposal on this? Can I write a project on this? You raised a question, you threw an hypothesis and argument, you are about to test it. At that point, you can have 2 paths. 1, go ahead and do experiment on your own, and also perhaps think of writing a proposal. Don't always write a proposal without yourself indulging in the experiment. If you indulge in the experiment, even if you fail, if you have a question and an argument, and even if you fail, indulge in an experiment, you will realize what are all the problems, and then perhaps write a project which is more meaningful and complete and without any gaps in it. So at that time, you can think of writing the proposal for a funding agency. Dr. Nataraj will come and tell you what funding agencies you can approach and how to approach, etc. So start the work and or write the proposal. I would say don't only start the work, I wouldn't say only write the proposal. It could be both. It depends on how you go about and think about it. Now let me come back to when to start with what kind of project, when to start with what kind of proposal. Now let me take on the x-axis amount of infrastructure and investment required for taking up a project. Some experiments or some questions that I have asked may not require any infrastructure at all, even if they require very less infrastructure, maybe a petri dish or some travel cost, etc. So let us call them as less infrastructure, less investment. And then some may have lot of requirements, high investment and high infrastructure. In between them falls the modest and moderate. Similarly, you will see that there is another dimension you can take. The other dimension is the amount of time and effort you have to spend on. If you have thought of a question that requires your lifetime, you better not perhaps attempt that, isn't it? But if the thought of a question is very simple effort, like for instance, Ganeshaiah, it's not a very difficult thing. So simple time and simple effort, so that could be one axis. And sometimes you need lot of effort and lot of time. So you see that there are some 9 sets of projects that one can think of. Among these 9 sets, I would say don't attempt them. What are they? Less investment, of course, but the amount of time and effort you have put in is so much that you do not want to do that because you are very young. You want to now take up those experiments which are, of course, require less investment, but not requiring too much of your effort. If you want to spend your entire lifetime, this is not the time for you to take up that. Will you agree with me? So don't take them up. And then don't also take them up. What is that? High infrastructure. You need a spectroscope. you need HPLC. But the amount of effort you want to put on that is very little. 5 samples you want to do. You don't need to now write a proposal for that. You can now pay someone and get it done wherever that instrument is available. So don't attempt them also. What you have to attempt is actually start from number 1, that is here, and move along this line. I don't say don't go into them, but that is the way you would like to proceed. Always at a young age, start with that very starting point, that is this cell. What is that? Less investment, less effort. What is that I can do? Among the questions that you might develop, focus on them. In other words, when you ask the question of what kind of projects and when to take up, start with very simple, low cost or no cost and zero investment or less investment projects. Okay? And build your own credibility with the small investment, very simple effort. You have now addressed a very interesting question. You have come out with a very interesting results. You have also published 1 or 2 papers. Then you can say, look here, I can also now work. I can also now go and look for proposals and money and ask for the funding agents. From there onwards, move on to build your group. Join 2 or 3 of your colleagues or network with other labs who are working in the same area. So build a group of your own and then establish the team's credibility by putting few more experiments and few more proposals, and then move on to large demanding projects. If you in the very beginning only say, I want to do on nanotechnology, give me 1 crore rupees, no one will give you. You have to establish your credibility. You have to build your team. You have to establish your team's credibility. Then people will automatically look for you. Aren't you now looking for any better large projects? They will ask you. And then you can now look for, okay? Don't ever look for huge projects in the very beginning. Now, when I say look for very simple projects that are less time consuming, what kind of projects? Let me now give you 1 or 2 examples of what kind of projects you can now look for. And don't ever think that any question is not worth answering. For example, now I am an old man. I don't know the performance of Kohli or Yarappa recently who scored 100. Rohit Sharma, but much more recently someone scored 100, no? Gill, Pant, who is that? Yashasvi Jaiswal, I love him. He seems to be now really emerging, correct? Yeah. But I am 69 years. I only know Tendulkar well, better than that. So I would like to know, in fact, I asked this question. Whenever Tendulkar is playing a 2020, a T20, and let us say India requires 40 runs. How many runs? Tendulkar is still playing. The question I would like to ask is, can Tendulkar win for India? No. Why, madam?
Why? Because at that time when he was playing,
Yeah. Oh, it was there. No, madam, it was there. No, 2020 was there. 2020, Tendulkar has paid. Yeah. No, it's okay. Okay. Who is your pet player? Rohit Sharma. Okay. I need 40 runs for India to win. Rohit Sharma is playing. Can you bet on him that he is going to win the match? Without. May I know your name? You are from? Bangalore. Vasavi seems to be a very conjuice lady. She wants to bet but not spend money on that. Very good, madam. How many kids you have? There also she is conjuice. She is very careful in that also. Good, madam. I also have only one daughter. But I am not conjuice. So, anyone can ask this question. Even youngsters can ask the question. India needs 40 runs. Jaiswal is playing. Can you now win? Right? Generally, people now bet if there is a big fellow playing like Jaiswal or Kohli or Virat Kohli or Rohit Sharma, isn't it? They do. But let us ask this question. Can these people really win a match? Isn't that a question? Can they win a match? You are actually now producing the data that would perhaps answer millions of people are losing their bets, isn't it? Isn't that a question to answer? So I exactly did this. How did I do? I first of all collected the score, average score that they are known to be now. Okay. So how much is the average score of Tendulkar? Sorry, this I am confused with this. Yeah, you see, his average is 52. How many score? I mean, how many runs we need? 40. So his average is more than 40. So can I say he perhaps can score? I am afraid. Answer is not so simple. That's where you have to develop the arguments very properly. So let us go ahead and look at what is that makes a player winnable. Number one, of course, mean score. If the mean score is very high, he is probably capable of winning. But will it ensure? Will it ensure? If the average is 60, will it ensure that he is capable of scoring 40 for me? Will it? ಯಾವುದಾಗಿಲ್ಲ ಜೋರಾಗಿ ಹೇಳ್ರಿ. ಒಳ್ಳೆ ಮದುವೆ ಮನೆಗೆ ಹೆಣ್ಣು ನೋಡಕ್ಕೆ ಹೋದಂಗೆ ಒದ್ದಾಡ್ತಾರಲ್ಲ ಹಂಗೆ ಹೇಳ್ತಿರಲ್ಲ. ಎಸ್ ಆರ್ ನೋ? ನೋ, average is not enough. And so, what is that we need to now look for? Sorry, there is some problem here. There is some problem. I went ahead. ಏನೋ ಆಗೋಯ್ತು ಇಲ್ಲಿ. Yes. So, in other words, it is not only the average that we are looking for, something else we have to look for. What is that? Let me tell you. Average score, let us take on the x-axis. So, we want a person to score very high average, correct? But on the other axis, we have got what is called variation of his score. 52 is the average of Tendulkar. What is the variation around that? You might also become out for 0 very next ball. You will lose your bet. Or you might score even 45. You will win your bet. So what is the variation? If I want Tendulkar to win, not only he scores 52, which is more than what I require, the variation around 52 must be less or more. Less, isn't it? Variance should be very less. In other words, along this line, we want those who fall here, but along this line, we want those who fall here. Agree? We want high average, low variance. In other words, we want people who fall in this category, and if there is Tendulkar or Rohit Sharma or someone falling in this category, I can say maybe he will win the match. Correct? Correct? Let us see, where does Tendulkar fall? I exactly did that. You go to websites, it's available in the websites. Get all the score of each of this fellow, find the average and find the standard deviation, plot along the average x-axis, variance along the y-axis of all the people. What did I get? Remember, what are we looking for? We are looking for someone who falls in this area, in this space. Okay? What is that I get? I get this. X-axis is the average performance, y-axis is standard deviation. You see, all of them are on this. Is there someone here? Is there someone there? No one will win the match. So, you cannot now bet even if the average is 80 of Tendulkar or of Rohit Sharma, madam. Don't bet on it. Even without money also, don't bet on it. ಆಮೇಲೆ ನಿಮ್ಮ ಮಗ ಕೇಳ್ತಾನೆ, ಅಮ್ಮ ನೀನು ಸೋತಿದ್ದೆ ಸಾಯಂಕಾಲ ಹೋಳಿಗೆ ಮಾಡ್ಕೊಡೋದಂಗೆ ಅಂತ. So, you do not have anyone who actually falls in this area. In fact, look at Tendulkar. His score is 52, but his standard deviation is also 52. He is as variable or unreliable as his high score is. In fact, I can make another statement. Sachin is only as unreliable as Kumble is around his score. Kumble's score is 5, but around his score we can rely on him scoring 5. But if you want to now rely on Tendulkar for 50, no, you will not be. Well, you see you have an answer. Tendulkar cannot be relied upon. I don't say I won't win. Cannot be relied upon for betting to score what I want. What I want is 40 runs or whatever it is. Are you all with me? Now, ಅಯ್ಯೋ ನನಗೆ ಗೊತ್ತಿಲ್ಲ ಮೇಡಮ್, ನೀವು ರಾಹುಲ್ ದ್ರಾವಿಡ್ ಲವರ್ ಅಂತ. ಓಕೆ, ರಾಹುಲ್ ದ್ರಾವಿಡ್ ಇಸ್ ನಾಟ್ ದೇರ್. ಸಾರಿ, Rahul Dravid is not there. Oh my God. Oh. So some of you can do it now. All this data is available in the website, and go and look. Incidentally, this paper was published in Deccan Herald. There was a lady called Manjula at that time in Indian Institute of Science. She was a scientist. She was also writing for Deccan Herald. She came to me and I was doing this and she asked me, what is that I am doing? I told, and she told, can I now write on this? She went and wrote. I did not write the paper. She collected all this material and she went and wrote that Ganeshaiah is doing this, Ganeshaiah is doing that. And so she declared to the world that don't believe in Tendulkar winning the matches. Anyway, so like that, you can perhaps now do wonderful work even on simple questions. Now the next question. If that is so, what is that? If Tendulkar is only as unreliable as Kumble is around the score, why is that Tendulkar gets so much amount of money and not Kumble? The answer lies in the next slide. This slide shows the performance of Tendulkar. How many times he has become out for 0, how many times for 10, 20, 30, etc. Median is 30, mean is around 50, and you see, more often he has become out at around 10. More often he has become out around 10 and also several times 0. But why he is so famous is that occasionally he hits a century. Of course, he has some 49 centuries or so, but that is occasional with the length of the number of innings he has now played. So Tendulkar occasionally scores these centuries and so Tendulkar is famous. Okay? Any questions on this? So that is a kind of simple experiment or simple test and simple questions we can. You might wonder, what is there science in that? Please note, any question answered using data and analysis is science. I'll come to another similar question that you can now perhaps look at. Now, in other words, you see that if you come back to, there are number of these kind of simple things one can work on. Look at this right hand side bottom corner, Nature, 1998. You know Nature journal? It's the top journal. In that there was a small article in 1998. What is this article? This group of scientists studied temperature of the earth along the week, Monday, Tuesday, Wednesday, Thursday, Friday, Saturday. Take the average of that and find out when the temperature is high, above the average and below the average. Surprisingly, what they found was, look at this, Tuesdays and Wednesdays temperature is more than the average of the week. Mondays and Saturday, Sunday temperature is less than the average of the week. Why could that be? Madam, please stand up. she is the culprit. You know, Friday means they now get off and all software people now will be in the bars and not in the office. So Saturday, Sunday, no vehicles go out. They don't move at all. Lot of burning of the petrol is not there. Monday, they'll be still sleeping in the hangover. 50% of the people don't go and some 50% go. But as the week becomes Tuesday, Wednesday, the boss will be scolding. He has not concluded the coding. And so they now take. So the vehicle movement will be very high on the Tuesdays, Wednesdays, Thursdays. So temperature rises. Isn't that a simple thing? It came in Nature. So my friend Umashankar, he wanted to see if it is true in Bangalore. One of our own students did that work. His name is Srinivasan. Look at Bangalore. It is not exceptional to only Western countries or otherwise, even in Bangalore. And that's why I see poor population today in this hall, because it is Monday. It was your wrong choice. Have a workshop on a Tuesday or Thursday, there will be 25% more people coming here. Some of them are still sleeping or they have not come back from their home when they went on Friday. Pardon me? On? No, it's okay. So, I mean, in other words, you see that the climate of Bangalore is affected by the psychology of the people. So, you look at the implications of all these questions and answers. The psychology and the working psychology of the people affects the climate of Bangalore. And so, if you are very intelligent, don't get out on Wednesday, it will be very hot. If you are intelligent, you get out on Saturday, Sunday, it will be cooler. Okay? Oh, she's very happy now. Shall I? Now, these are the simple things. When I say simple things that require very less investment, isn't it? It requires very less investment. You simply have to go to websites and look for the data. Believe me, mining the data can be the most exciting part of the work anywhere that you can think of, in any field. There is so much data lying which we can perhaps trap and work on. So, what and when to now work on, I told you that that is the first to approach on, and I will now give an example to show how the second kind of work can be done. What is the second kind? Little more effort is required, and some more time and effort and infrastructure also perhaps is required. Now, that's where I will answer your question. What is that? Why do plants have medicinal properties? In that, let us take this. Laxative is a medicinal property. Plants have laxatives. We use them to now clear our bowels and make sure that early in the morning we don't have any trouble. Why should plants have that? What is their problem if I have the problem in my bowels? Isn't it? Right? So, laxatives are the chemicals that induce bowel movement, but plants don't have bowels. So, the question is, why should they have laxatives? I have put this question in red again to emphasize the following. See, we have taken some of these things as obvious, that a plant has a medicinal plant. But it is not to be taken as obvious. You have to ask a question, why the hell a plant has a laxative that is going to help me? So, that is the beginning of the research. So, why should plants have laxatives? Now, if you start thinking carefully, and I told you, these are the kind of questions where you need to now spend extra amount of, thinking, discussing and all. So, plants disperse their seeds for better establishment. You all know that. Okay. And what are the ways in which they disperse? They disperse by wind, they disperse by explosion, or they disperse through animals, and they disperse also through water. Among all these, remember, which are the ones that are connected with the plants in terms of bowel movement? Is the wind connected to the plants with the bowel movement? What bowels wind has got? Animals, isn't it? So, animals and plants are connected somehow, and animals have bowels. Could there be something there? So, in other words, if you don't look carefully, some species pack their seeds in fruits. They attract birds and animals so that they eat fruits and disperse them, correct? And these animals have gut system. So, maybe we can link up the laxatives that plants have got with these animals that disperse the fruits and seeds, correct? Okay. Now, how can we still link it up? If you actually carefully now start thinking about, it turns out that plants attracts birds and animals to eat their fruits, but the plants do not want these birds or animals to keep their seeds in their stomach for too long. You know what happens to the seeds if they keep them for too long? Yeah, the seeds get damaged, isn't it? In other words, if you now look carefully, along the x-axis, let us take the passage time meaning the time the seeds are there in the stomach. And on the y-axis, health of the seed. Longer they stay in the stomach, what would happen to the health of the seed? Decreases. So, we will now draw a line. If seeds stay for too long in the gut, they are damaged. So, the health goes on decreasing, correct? But what is the interest of the plant? To disperse. What happens to dispersal if they are now staying in the stomach for too long? The birds go far away from the mother plant, and so dispersal increases. So, longer they stay in the stomach, dispersal increases, but longer they stay in the stomach, health goes down. So, in other words, if I now write on the y-axis dispersal advantage, dispersal goes on increasing. On one hand, one benefit goes on decreasing, but another benefit goes on increasing. A compromise between the two, a trade-off between the two is what a plant wants. And so, good health, if they stay for too short, they are not dispersible, and so they have to now keep them in the stomach for long. And so, as a compromise between the two, you see that the fitness of the seed or survival of the seed and dispersal increases and then decreases after some time. It decreases because it gets damaged. It increases, the fitness or survival or the advantage increases because dispersal is increasing. Are you all with me? So, if you now look at the advantage to the plant or to the seed, there is certain time after which seed should not stay in the stomach. The animal should throw it out. How can you tell animal, oh, throw it out at 2.5 hours exactly? Plant cannot dictate that. What do? I pack my seeds with some laxative so that as it eats, after some time, laxative takes care of my purpose. Okay? And that is, in other words, the seeds should stay for only certain optimum length of time. They should be thrown out only at that optimum time. If they retain for long, the seed is damaged. If they are dispersed early, thrown out early, dispersed is affected. And so, they can achieve this by packing laxative in the seeds. Are you all with me? You all agree with that? Now, we have an argument. We raised a question, we have an argument, and now to the experiment. How can we test this? Let us say there are these dispersal kinds. What are they? Animal dispersal, wind dispersal, and explosive dispersal. Among all these, who require or what kind of plants require laxatives? Animal dispersal. So, one of the predictions we can put is that animal dispersal species should have laxatives more than wind or explosive dispersal. Agree? Now, we have the test. Do we have to run behind animals now? No, you only have to go to literature. Go to the literature, look for where plants have, which plant have got laxative, what is the dispersal mode, your problem is resolved. But you need some work to be done. And this is where we did some work. One is data on plants possessing laxatives. Where do you get that? Materia medica is there. Go and look for wherever they say this plant material can be used as a laxative, take that as laxative containing. And dispersal mode of the plant. There are a number of sources. Our major source was this Ridley 1930. So, list down all these characters of the plants and you now test for the association. Let us not bother about the details. If any of you want, you can go and look for this paper in current science. What is the result? Here is the result. Sachin, how much time I have? Huh? 15 minutes. Okay. So, here is the result. If you observe the total plant community, about 40% are wind dispersed. If you take all the plants, 40% of them are wind dispersed and about 35% or so are passively dispersed and about the same 35% or so are animal dispersed. Let us say our hypothesis is wrong. What is our prediction? That animal dispersed species should have laxatives. Let us say that is wrong, that all plants equally have laxatives. Then the number of plants that have laxatives should also be distributed in the same proportion, correct? 40 in wind, isn't it? Now, when we actually got the data, this is the data. I don't need to know TACOM. 60% of the animal dispersed species have laxatives. How many we expect? Only around 30%. Whereas, the wind dispersed, we expect 40% of them to have laxatives, only around 10 or 11% have. And in the passive, there is small difference there. In other words, predominantly or very strongly, our prediction is right. What is that? Animal dispersed species have laxatives for packing their seeds and sent out through animals so that they are now delivered at the exact time that the plant wants, so that the fitness of the seed is now increased. Okay? And what else I have here? So, a high proportion of animal dispersed species have laxatives. And if you want to explore plant with laxative, now the implication. Let us say you are a medical doctor and you want to now investigate or find out a new plant that has a laxative. Where would you go and look? You look for in a wind dispersed plant? No. You would rather go and explore that in the animal dispersed. There are 20,000 plants with us. There are only 30% of them which are animal dispersed. So, you only look for them. Otherwise, you have to explore in all the 20,000. So, this kind of work has other implications. So, this is in other words, pharmacognosy, looking for medicinal value. By what? Not by looking at animals, but by your deduction process, by your own inferences. Okay? So, there are different ways in which you can actually discover medicinal plants. This is the kind of modest work and modest infrastructure. There is no infrastructure required here, but you need to now get the data and work on that. Now that's an example. Here is another example. Again in nature, I don't think I have the reference brought in, but nature. Someone, a chemist has produced this in nature. What is that? He has listed all the chemicals containing carbon, some thousands of them. And when listing, he has found that somehow chemicals containing even number of carbons is more than the odd number of carbons. Are you with me? Look at this graph here. That red marked ones are all even numbers. So the proportion of that is chemicals with the 2 carbon, 4 carbon, 6 carbon, 8 carbon, 10 carbon, 12 carbon or 120 carbon or 140 carbon is more than 139 or 119 or 7 or 5 or 6 or 3. Why? Incidentally, if you carefully see this paper, you will see that this is not known why. 7 million organic compounds have been screened, have been now taken for this data. Even today, no one knows why the even numbered carbon atoms are more than the odd number carbon atoms. Now, this is only a question raised here, not answered. There are so many such things one can now look at. I think I am coming to the end. That is the second one that we discussed. The third one, think of mega projects only when you have strong base. I wanted to show some data of this. Sachin, when you have some time, show the Western Ghat data. I will not perhaps go into this because the time is over. I will perhaps come to, I will skip all this. Okay, I'll just show you. See, once you have built up the team, once you have now put a base, you can now look for mega projects. One among such mega projects we did was mapping the entire Western Ghats for the plants. I will not go into the details, but let me just tell you. 7 teams, each with 5 people in that, worked for this for about 7 years. You would not like to wait for such a long period if you are a very young man. But I could afford to wait because I had spent almost 50 years of my age. And at that time, I now started working on these mega projects. So as a result of it, we now came out with the data on the Western Ghats for the species discovery and all. I'll just show you a small sample of this. So this is Western Ghats, as you know. If you want to know what we did was, we divided the entire Western Ghats. It's not visible here. No, it's not visible here. Why is that? Oh, as usual, same problem. Yen madbeko? The program is not coming onto this. Escape padadu. Exit. How do I do that? I can go here. Can I? Western Ghats. Okay. So what you can see here is that the entire Western Ghats is around 1,600 kilometers and 1.4 lakh square kilometers was divided into grids of 40 square kilometers. 40 square kilometers, 6.3 kilometers this way, 6.3 kilometers this way. In each of the grid, people walked and observed the trees and herbs and shrubs and epiphytes and all the plants, and the data was gathered. For the first time in the entire world, we have the finest data ever possible collected, and I'll just give you a small sample of what can be done. The reason I'm showing the here is not to propagate or to give an advertisement of a commercial break alaidu, to show you how mega projects will look like and when perhaps we can look at these mega projects. Okay. So this is the database, and Sachin is very much a part of this. Now, what I told you, the entire Western Ghat was divided into 3,331 grids, and that is the map. Suppose I want to now know what is available, let us say, in this part, which is actually a Karnataka part. Now, mark the grids that you want to now know the data on. What you see is that it labels the grids, it tells you what are all the trees available in those set of grids. And if you want to now know the shrubs there, go and click the shrubs, you will get the shrub list, herb list, or epiphytes, or total number of plants. Now you want to now look at any one plant, let us say acacia chundra, select that, and you will have the images. You have line diagrams, herbarium diagrams, photographs, acacia, chundra. I don't have time, we can now put on each of them and expand them on view. But more than that, you also have the synonyms for that and common names, what it is called in Hindi, Kannada, Malayalam, etc. and distribution of that. Based on the data that was collected, we now can develop this kind of maps. Where is acacia chundra in the entire Western Ghats? Where is that? It's available there. All the marked grids have acacia chundra. How much? This much. If there is a blue color, it is 15 to 127. You know, the scale can be changed. 127 plants are there in that. If it is that, whatever that color, 3 to 5 plants. If it is not there, there are no plants. If today or tomorrow, if you are a medicinal plant extractor and if you want to go and harvest medicinal plant of acacia chundra, you know where to go and look for. And where is it abundantly available? Which of the states is more rich in terms of acacia chundra? You see, Pune and southern parts of or northern parts of Kerala and southern parts of Maharashtra are more rich in acacia chundra. So if a company is now launching any product on based on acacia chundra, they would know where to go for. And if you are a scientist, they'll come to your office and they'll pay you any amount of money you claim, and they'll be able to give you that. Okay, as I told you, I don't like to now go into this for too long. This is only to show, and let us say you are from, madam, you are from which Tamil Nadu? Okay, Tamil Nadu, which district? Krishnagiri. No, but that's not Western Ghats. Anyone from Western Ghat area? No Western Ghat area? Yeah? Coimbatore. Okay, let us see. Tamil Nadu. Coimbatore. In Coimbatore, which taluk you want, madam? Pollachi. You now click that. The data will tell you. It may take some time, but I will assure you that it will tell you, it lists down all the plants available in Pollachi of Coimbatore district. It takes some time. The data is very huge. It has to cut down. Or any plant you can look for. Like this database is now available for Andaman and Nicobar, Western Ghats, Eastern Ghats, and partly Northeast India. And a Vindhya range project is going on by someone else. These are the mega projects. They are for the nation. And when you now develop these projects, note that you won't gain much. You are contributing to the country. But you will have the satisfaction that you have done wonderfully, useful projects. Yeah. Not losing. If you want endangered species, where it is, I can tell you where they are.
Yeah, but if they invariably go out of that space for some reason, somebody shot somebody.
We will not know. This data has been done once. In fact, Sachin wanted to do a second survey of this.
Does it get mapped to this layer?
No. This is a ground truthing. As I told you, the 7 teams dividing the whole Western Ghat into some 5 areas. They have gone to each of this area and they have now located this, got the data. And in fact, Sachin wants to now repeat this work. It's a huge project.
Carbon footprinting, biomethylation process.
We
From the forest, they get those pockets of wherever.
We have mapped the carbon stock of the entire Western Ghats, madam, based on this data. There is a paper on that. If you want, I can now send you that paper. Along with the NRSC Hyderabad, the ISRO people, we have now mapped that also using this data. So a number of things can be done. Now, for instance, you want to now know where are the endangered plants, where you want to conserve them. Or you want to now know where is the biodiversity of Western Ghats rich. You want to conserve them. You can identify all these areas. So these are things that you would like to do when you are little old and perhaps not immediately. Let me skip all this. Okay. I have here as the last part some questions. One can go on and asking these questions. Just as an example, I have given some. Why do birds bring fresh twigs? I already answered. How do ants forage complex environments? You look at a small ant that will be going onto a complex tree, finds the nectar and goes back to its nest. How does it know? Namige kashta Bengaluru olgade hogbittu Malaysia menda vapas barbekare dari tapputre. And the ants in such a complex environment, they can find their way back. How can they do it? Now these are, one can, and who contributes more to Kannada literature? I'll come to that a little later. Now, things of this kind. Why are there 7 notes in music? In music, why? Have you questioned why there are only 7? No one prevents you to add one more letter, say cha, sarigama padanisa, cha no hakvodalla, yak hakilla. Or when did Kannada script evolve? Is there weekly cycle in climate? I think I showed you that. So any of such questions can be asked and each of them can develop into what I'm going to know, spend the next 3 to 5 minutes is this question, who contributes to Kannada literature? The question came to us in our house. I think Rajini was there in the house at that time. My brother has a son who got admitted to engineering. So he came home to tell me that he has got admitted to engineering and he wanted to get my Ashirvad. I am a Buddha, I am having the beard, so I am old man, I can now wish people. He came and then after some time he went up to my first floor and he was searching for something in the first floor. I thought he is looking for some of my novels, Kannada novels. So I shouted to him and told, "Lo, ninge novels bekadre balagade nodu anta." My brother told, "Avanu Kannada odalla. He doesn't read Kannada." I told, "How is that? Didn't he take Kannada as a subject?" He took Kannada as a subject. He has scored 99. Then how is it he can't now read Kannada? Our amma previous day ಓದಿ ಹೇಳ್ತಾ ಇದ್ರು, ಅವನು ಕೇಳ್ಕೊಂಡು ಎಲ್ಲ ಅಲ್ಲಿ ಟಿಕ್ ಮಾಡಿ ಬಂದಿದ್ದಾನೆ. This is how it happened. I started, wondering at this. In the same family, there is a fool who writes Kannada novels and there is an engineer who can't even read Kannada. What irony is this? My brother told, no, he will become a novelist in Kannada. I asked why. If you look at all the Kannada writers today, they are all software engineers. He has now joined software and he will also become a Kannada. You are all with me? There was a student of mine called Harisha. He asked me, "Sir, is it true that people from software write more in Kannada than others?" I told, "Why don't you test it?" And that's how the question started. So he went about, and I'll simplify the whole story here. I don't like to go into the details. He went about. So the common belief is that software community are turning to Kannada reading and writing. So is it true? They are contributing to growth of Kannada in the recent past. Is it true? That is the question we asked. And what he did was, he went to the data. Publishers have the list of books that they publish, Kannada publishers. He enlisted all of them. About 175 authors he enlisted and he could get the degree, whether they are BSc or BE or doctor or BCom, etc. Once that data was obtained, all 175 he could not get, only 123 he got. And then he also now started developing what are the subjects that people generally take when they go to the degree classes. Now, if you look at 2000 to 2015, if you now take this 15 years, about 42% of them have gone to BA. About 15% have gone to BCom. And science, about 20%. BE, about 17%. Medicine, only 1%. Competition, no? Severe competition. So this is the percentage of the degree program that students go into. If all of them are contributing to Kannada equally, what is that you expect? You expect in the same proportion, isn't it? Correct? Okay. Now, what is the data? Data is as follows. Approximately true, but look at the medicine. Only 1% of the people go to medicine, but there are something like 12% of the writers who belong to MBBS. And engineering, 17% of the people go to engineering, about 24% of the authors are from engineering. So MBBS and engineering students are contributing, in fact, more to literature. To cut the story further short, it turned out that for every one that we expect, about 12 extra MBBS people are writing. So 12 times more authors are there than expected in Kannada who are from MBBS. And if you now look at engineering, that is what is expected if it is 1-to-1, and engineering about 1.4. So 1.4 times more engineers are writing in Kannada than what is expected. I mean, just trying to give you examples how we can now start working on. This is not science, this is literature, but you are using scientific methodology for that, and we published that in Sudha. We could have sent it to Current Science, but Sudha wanted this, and so we wrote it to Sudha. So this kind of work also can be now done. Eventually, I will not go into the reasons why and all. So final slide, few remarks. World is a fantastic laboratory. Don't always say that our principal has not given me PCR machine. No, you don't need. World is in fact a fantastic laboratory. We have hardly explored it. Living world in particular is a golden laboratory in my view, and there are so many things that we can now look at it. There is more unknown in the living world, as a matter of fact, and so wonders can happen around us. Catch any of those wonders, you will be able to do some work, and each of you can do wonderful work. Thank you very much.